Hello and welcome to the course Basics of Experimental Design for Cognitive Psychology. I am Dr. Arik Verma from the Department of Cognitive Science at IIT Kanpur. We are starting the week 8 of the course where we are continuing to discuss about different methodologies that utilize experimental research designs. In this week, I will begin with discussing about event-related potentials in today's lecture and a couple of more lectures. Now, the ERP technique or the event related potentials technique, it provides a powerful method for exploring the human mind and brain. The technique involves the measurement of electrical activity in the brain contingent to a presented stimulus or an event, and it provides an index of the temporal processing of that stimulus in the brain. So, that is basically why it is considered very important. Whenever you are presenting a stimulus to the brain, you want to know what is happening post presentation of that stimulus, or in some cases how is the brain preparing for receiving a particular kind of stimulus, acting on it while the stimulus is still presented, and what happens even after the stimulus is gone. So all the temporal aspects of cognitive processing are best studied using the event related potentials technique. Now, before we dive further, let's have a brief historical review of how does this technique develop. So, the ERP methodology, it seems to have been discovered in the early 1930s and has now progressed over ensuing 100 plus years almost. One of the first interesting events was in 1929 when Hans Berger reported a remarkable set of experiments wherein he showed that the electrical activity of the human brain could be measured by placing an electrode on the scalp, amplifying that signal and plotting changes in the voltage over time. So that is basically the first time people discovered that electronic activity in the brain can actually be measured, and this is the contribution of Hans Berger. This electrical activity that you could record from electrode on the scalp was referred to as an electroencephalogram. Over the following years, the EEG or the electroencephalogram proved to be very useful in both scientific and clinical applications. However, if you look at it, the EEG is a very coarse measure of brain activity and it cannot be used in its raw form to measure the highly specific neural processes and that are studied in cognitive neuroscience, the kind of questions you want to ask. So there is always electrical activity in the brain and it's a summation of a bunch of processes that are going on. But if you want to know what is happening in the brain contingent to a specific stimulus, then you cannot work with the raw EEG form. You will have to devise new methods. You have to devise a more sharper way of analyzing this data, which is basically what we're going to talk about. As I was saying, EEG represents a mixed up conglomeration of dozens of different neural sources of activity. It is difficult to isolate individual neurocognitive processes from the electroencephalogram. Within the EEG, for example, there are neural responses associated with specific sensory, cognitive, and motor events, and it may be possible to extract these responses from the overall EEG by means of a simple averaging technique. These specific responses that you can gain or that you can isolate, which are contingent to specific actions or specific events, are referred to as event related potentials. That is, these are the electric potentials that are related to specific events or specific stimuli which the brain might be processing. Now, one of the earliest ERP recordings from humans was first reported by Pauline and Hallowell Davis in the years 1935 and 36, when the researchers were able to isolate clear ERPs on single trials during periods in which EEG was relatively stable in a quiescent state. The first ERP waveforms were published by Galambos and Sheets in 1962. Now, it seems there was a bit of a stoppage in ERP research during the years of the World War, and it took some time to come up. So the modern era of ERP research actually begins in the year 1964 when Walter and colleagues reported the first cognitive ERP component, an ERP component that was contingent to neural processing. That component is referred to as the contingent negative variation or the CNV component. Now, how did they come about this, or how did they discover this? On each trial in their study, subjects were presented with a warning signal, just like, let's say, a click sound, followed by 100 or 500 or 1000 milliseconds later by a target stimulus. So there is a warning signal, and after that the participant is expecting a stimulus, and that target stimulus happens around 500 or 1000 milliseconds later. In the absence of a task, both the warning signal and the target elicited the sort of sensory ERP response that would be expected for these stimuli. However, if the subjects were required to press a button on detecting the target, a large negative voltage was observed at the frontal electrode sites during the period that separated the warning signal and the target. So, between the time the warning signal has come and the target has not yet appeared, and the participants are expecting a target stimulus to appear, there is this negative voltage which is being observed in the frontal areas of the brain, in the frontal electrode sites. This negative voltage was referred to as the contingent negative variation, and it was taken to reflect the subject's preparation of the oncoming target. So, the subject is preparing to sort of deal with this oncoming target and process it. So, this new finding encouraged many researchers to begin exploring cognitive components, because now it showed a window that there is electrical activity in the brain, which we were calling the electroencephalogram or the EEG, can actually be analyzed well to slice out specific electrical events in the brain that are tied to specific events, that are tied to specific cognitive operations that the brain does. So, another important step in the same direction was the discovery of the P3 component by Sutton and colleagues, and here the researchers created a situation in which the subject could not predict whether the next stimulus would be auditory or it would be visual, and they found that the stimulus elicited a large positive component that peaked around 300 milliseconds post the presentation of the stimulus. This positive component that peaks 300 milliseconds after the presentation of the stimulus was named as the P3 component or the P300 component. This component was found to be much reduced when the conditions were such that the subjects could predict the modality of the stimulus, and the difference in brain responses was attributed to the difference in the predictability or the unpredictability of the stimulus. So, if the stimulus was unpredictable, then the P3 component was slightly larger, as opposed to when the stimulus was predictable, that it was going to come in an auditory modality and the subject is able to predict that. Now, these two were the first cognitive components that could be isolated from the EEG recordings, and after these and similar findings, researchers basically lapped up the ERP research paradigm through the 1970s and in 80s, and they took upon that to investigate the general exploration of questions that are required in cognitive neuroscience. Now, let us explore some examples of how the ERP methodology has been used to investigate cognitive processes. Look at this. First is the classic oddball paradigm. I am sure everybody knows that in a classic oddball paradigm, a particular stimulus is presented again and again and again, and very rarely another stimulus is presented, which the participant has to detect and respond to. Okay. So, in the classic oddball experiment, subjects viewed sequences containing 80% X's and 20% O's, and they had to press a particular button for X's and another button for O's. Each letter was presented on the computer monitor for 100 milliseconds, followed by a 1400 milliseconds blank interstimulus interval. So, a stimulus will come, and then there is a gap of 1400 milliseconds, another stimulus will come. Most of the time, that is 80% of the time, the stimuli will be X's and sometimes it will be O's. When the participant detects an X, it presents a particular button. When a participant detects an O, it presses a particular button. Now, when the subjects were engaged in performing this task, the researchers recorded the EEG from several electrodes embedded in the electrode cap that was placed on their scalp. The EEG from each site was amplified by a factor of 20,000 and then converted into digital form for storage on the digitization computer. So, you're basically getting the signal and then storing it for further analysis. Whenever a stimulus was presented, event codes were sent from the stimulation computer to the EEG digitization computer, where they stored it along with the EEG data. So, you're basically tagging or marking the EEG data with respect to what event is going to happen. Now, in each recording session, the researchers viewed the EEG on the digitization computer, but the stimulus elicited ERP responses were typically too small to discern within the much larger EEG. EEG was recorded at one electrode site from one of the subjects over a period of 9 seconds. The EEG waveform was first recorded from the PZ electrode site, which is on the midline of the parietal lobes, where the P3 component was found to be the largest. On a closer examination, it was clear there was a consistent pattern in the EEG response to each stimulus, which became even clearer when the continuous voltage was converted into a discrete set of samples for storage on the computer. So, when the signal was analyzed further, it started becoming clear that there was contingent electrical activity in the brain with respect to the stimuli. Now, the EEG was being recorded concurrently from about 20 electrodes placed in the 1020 system according to the protocol set by the American Encephalographic Society, and the system names each electrode using one or two letters to indicate the general brain region. Say, for example, F and P for frontal pole, F for frontal, C for central, P for parietal, and O for occipital, and T for temporal. So, these are basically how the names of these components are kept, or names of these electrodes are kept actually, and a number to indicate the hemisphere and a distance from the midline. So, for example, a lowercase z is used to represent the number 0, which basically indicates that the electrode was placed along the midline of the scalp. Alright, so thus F3 lies over the frontal cortex to the left of the midline, whereas FZ lies over the frontal cortex on the midline, whereas F4 lies on the frontal cortex to the right of the midline. So, this is again just some convention that we ought to be aware of. This is basically the setup. You can see that the subject, an electrode cap is placed on the subject. These are the active electrodes. There is this reference electrodes. There is the stimulation computer and monitor. All of these feed into the, go through the filters and amplifier, go to the digitization computer where the analysis is taking place. These are the different electrode sites, the 20 of them from which the data is being observed. Here you can see we have zoomed in this region of the particular signal, and this is basically how that signal sort of comes across. You can see this is the average of 80 X's and this is the average of 20 O's. You can see that there is a difference in the peaks that is observed contingent to X's and contingent to O's. Now, at the end of each recording session, the researchers were performing a simple signal averaging procedure to extract the ERP waveform elicited by the X's and the ERP waveform that was elicited by the O's. The basic idea that was that recording EEG contains the brain responses to the stimulus plus also other activity that may be unrelated to the stimulus, which basically therefore needs to be extracted to yield a consistent response by averaging all the trials which had X's and all the trials which had O's. To achieve this, the researchers extracted the segment of the EEG around each X and O and lined up these EEG segments with respect to the event codes that mark the onset of each stimulus. So, starting from when the stimulus was supposed to come till when the stimulus has passed, they carved out those segments and then they analyzed those segments. So, after this, these were simply averaged together, over the single trial EEG waveforms, creating one average ERP waveform for the X's and one average ERP waveform for the O's at each electrode site. So, this is what you can see at each electrode site. You get particular waveform for X's and another particular waveform for the O's. So, for the voltage at 24 milliseconds in the average X waveform was computed by taking the voltage that was measured 24 milliseconds after each X stimulus and averaging all of these voltages together. So, this is just a bit of a processing convention on how this data is cleaned. Any brain activity that was consistently elicited by the stimulus at that time will basically still in the average. However, any voltages that were unrelated to the stimulus will be negative on some trials and will be positive on some trials, and therefore they will cancel out during the averaging. The resulting averaged waveform basically will be consisting of a sequence of positive and negative voltage deflections, which we can call peaks and waves or components, and you can see here that they are named separately. So, you have P1, you have P1, N1, P2, N2 and P3. So, these are the peaks. This is the first positive peak, second positive peak, third positive peak and the first negative peak and the second negative peak. So, this is basically by convention how these waveforms are actually labeled. Alternatively, the number may indicate the latency of the peak in milliseconds. So, sometimes you will also come across ERP components that have the number which tells the latency or the time period of when that peak is observed. So, here we see P1, N1, P2, N2 and so on, but you can also have a component such as the N170, which basically says that this is a negative peak which peaks at negative component which peaks at around 170 milliseconds post stimulus. Alright. Now, components may also be given paradigm or function based names, such as sometimes there are components which are referred to as the error related negativity or the no go N2, which is basically observed on the no go trials in go no go experiments. The sequence of ERPs peaks basically reflects the flow of information through the brain, the temporality basically. So, how does the brain react first and how does it react second? So, all of these peaks P1, P2, P3, first positive response, second. So, in chronology, P1 will come first, P2 comes second and P3 comes later. So, the sequence of ERP peaks basically reflects the flow of information throughout the brain and the voltage at each time when the ERP waveform reflects the brain activity that was going on at that precise moment in time. Now, in the experiment that we have just seen, the infrequent O stimuli elicit a much larger P3 wave than the frequent X stimulus, a finding that has been replicated with thousands of previous oddball experiments. So, you can look here, the infrequent O basically leads to a much larger positive peak which goes over 10. The rare O's actually reflect a much larger P3 component than the very frequent X's. So, this is the finding, this is a critical finding in the oddball paradigm, and this is something that has been replicated and repeated across several experiments. More specifically, the P3 wave was also largest at the PZ electrode, but could be seen at all 20 electrodes. So, it was observed at all 20 electrodes, but it was found to be largest at the PZ electrode. The P1 wave in contrast was the largest at the lateral occipital electrode sites and was not present in the frontal site. So, this is again you can differentiate between which are the peaks, where are they appearing and what time courses they are peaking or not peaking. Now, here you can see that each ERP component has a distinctive scalp distribution that reflects the location of the patch of cortex in which it was originally distributed. So, each ERP component basically has a proper distribution that basically tells us the location of the scalp where it is coming actually from, where it is originated from. Now, this is about the oddball paradigm. Let us look at another very important ERP paradigm. These are just examples that we are taking to illustrate how the ERPs are collected and how they are able to tell us something about the brain. Now, another very interesting experiment is there which yields a specific N170 component, that is a phase related component typically peaking around 170 milliseconds after stimulus onset and is largest over the ventral areas of the behind areas of the visual cortex. Now, in a typical experiment, in a typical N170 paradigm, photographs of faces and other types of non-face objects such as houses or cars are flashed on a computer monitor while the subjects are passively viewing the stimuli. Okay. The X presents the time relative to the stimulus onset and the Y presents the magnitude of the neural response. You can see here the X is the time here and the Y axis is presenting the response in microvolts that is contingent to a particular response. Okay. In the scalp map is shown, the shading indicates the voltage measured at each electrode site during the time period of the N170 response. So, you can see here there are these different shading, they basically tell us the amount of voltage that is experienced at different sites when this particular when this particular component is isolated. You can see this one is the N170, it is a negative peak happening at around 170 milliseconds, peaking at around 170 milliseconds, you can see here, and this is happening in response to phase stimuli but not in response to any other kinds of stimuli. So this is notable because it is larger when the eliciting stimulus is a face compared to when the stimulus is a non-face object such as a car or a house or anything. The difference between faces and non-face objects begins approximately, it starts at around 150 milliseconds after the onset of the stimulus and peaks at around 170 milliseconds and then sort of recedes back, which basically would allow us to conclude that the human brain is able to distinguish between faces and other objects within a very short period of 150 milliseconds. The scalp distribution helps us to know that this is the same component that is observed in other similar studies of the N170, and it suggests that the N170 generator basically lies somewhere in the visual cortex, in the back part, in the ventral part of the visual cortex. Now, several researchers have used this N170 paradigm, and they have basically used it to address interesting questions about how faces are processed in the brain. For example, some studies have asked whether face processing is automatic by testing whether face elicited N170 is smaller when the faces are ignored. So, if the face is there and you're not consciously processing it, does the N170 appear again? So, that is basically what some people wanted to look at, and the results of these experiments actually indicated that face processing is at least partially automatic, but it can be modulated under some circumstances. So, when the faces are being actively attended, then the nature of the peak versus when the faces are partially ignored is slightly different from each other. Other studies have used the N170 to ask whether faces are processed in a specialized face module or whether the same neural processing is also used when people process other sorts of complex stimuli for which they have extensive expertise. For example, so some people have linked the N170 to the role of expertise as well, and therefore very interesting experiments have happened, and it has been shown that bird experts exhibit an enhanced N170 in response to birds, dog experts exhibit an enhanced N170 in response to dogs, and fingerprint experts exhibit an enhanced N170 in response to fingerprints. So, there must be some role N170 with regards to our expertise in processing faces. That is why it basically is elicited by faces. So, that is all that I wanted to talk about in this lecture. We have just initiated a bit on ERPs. In the next two lectures, I will take this discussion a little bit further. Thank you.